Good morning, everyone. We just wanna start so we can get you out of here before it gets too hot. My name is Akif Eskalen. I am a professor of cooperative extension in the Department of Plant Pathology. I am a mycologist working on the mainly grapevine diseases. I also work on the tree fruits and small fruits at the Department of Plant Pathology at UC Davis. Before I start talking points, I would like to acknowledge the research project colleagues and collaborators. First of all, I like to acknowledge my lab members, mainly Molly Arequin, who is the technician in my laboratory who has done most of the work in this trial. I would like to thank. And rest of my lab helped her getting all things done. I like to thank to Annika King-Hapvala. She is here. Philip Roshausen, Stephen Lindo, and those are our colleagues. That they are going to have time to explain how they are involved with this experiment as well. So today we are here to see some of the preliminary results to control or prevent Pierce's disease on grapevine. So I'm sure you know already, but the most common symptoms of the Pierce's disease are starting with the leaf scorching around the leaves. The reason for that, when the bacteria is introduced into the xylem, wherever that they are inhabiting, they colonize, they plug the xylem vessels by colonization in the xylem and prevent the movement of the water and nutrition up to that. Because of that, you start seeing the first symptoms as a drought or lack of the water because it's prevented by the bacteria. That's one. Those colors could be different between the red and white varieties. I'm going to show you in a second. On the red varieties, you can see the little bit reddish color around the scorch of the leaves. That's very common. When it comes to white varieties, you don't see that color on the scorching areas. Later then, that leaf can drop off and then live with the stick or petiole of the leaves. We call it matchstick. That's another common symptoms. If you want to compare with the fall leaf removal or drop off, it leaves from the entire petiole or the leaves. When you have this matchstick or the petiole sticking on the shoots, that's another common symptom of the Pierce's disease. Another common symptom is the different color, light and green color or brown color on the shoots when shoots start lignifying late in the season. So we call them island the symptoms. That's another common symptom. And fruit shriveling is another common symptom that you are going to see in this trial. So it doesn't have to have all these symptoms in order to have disease. The plant could show either one of them or more than one or all of them. Those are the common symptoms of the things that we are going to see right now. So those are the common or general information that I would like to share with you. So what are we doing right now in here? So we have a project that was funded by Pierce's Disease Board last year. So this is our first year of trying some of the available biopesticides to prevent and control the disease of the Pierce's disease. With this, our trial here is that a little information about this is Cabernet Franc. It's 12 years old vineyard that hasn't had any history of Pierce's disease before. And in this trial, we tried 10 different treatments, including, you can see from the advertisement here, one of the bacteriophage, which is a virus that attack the bacteria. which Anika is going to talk more about it. That's one treatment. The other treatments are biocontrol agents that were developed, found by our colleagues from UC system. One of them is Steven Lindo. He is here. He's going to talk about it. And other one is the Philip Rosazon. He is here. He's going to talk about his the biocontrol agents. So as a UC researchers, we wanted to find out all the available biopesticides and then apply here and then challenge them by artificially inoculating the Xylella fastidiosa to see how they perform to prevent and control the diseases. We usually organize this kind of field days after our experiments. It's very unusual to have first-year results. When we saw the results in July and in August, we got excited, and then we wanted to share and show this information with you after the first year. Again, we are just sharing preliminary data from this trial that we have done. So what we have done as a trial here, we have a healthy control. There's a place that grapevines that we haven't applied anything. That's our healthy control, okay? We have a disease positive that I'm gonna show you in a second, that we artificially inoculated with the Xylella fastidiosa by drop puncture and then let them have the disease. We have phage application, biocontrol 1, 2, 3. We have 3 different biocontrol agents. And we also wanted to combine these biocontrol agents with the phage to see if they have a combined effect to control these diseases.